Good morning, all. I'm Paul Snyder, Vice President of Healthcare for Right to Market. We are a digital marketing and public relations agency and the marketing partner for this series of Pediatric Tech Talk webinars on behalf of the Georgia Tech Children's Healthcare of Atlanta Pediatric Technology Center. Thanks to all who have joined today. Please allow me to especially thank and introduce our panelists. Leanne West is Chief Engineer of Pediatric Technologies at Georgia Tech and Chief Innovation Officer of the Children's Healthcare Atlanta Pediatric Technology Center. She is also President of the International Children's Advisory Network. Dr. M.G. Finn is James A. Carlos Family Chair for Pediatric Technology and also Chair of Chemistry for Georgia Tech. Thank you all for joining us. Leanne, I'm going to let you and MG just take it away.
Okay. So MG, what are we here to talk about today? What does your research focus on?
So we're here to talk about a subject that spans basic science to clinical application. And the subject is the use of viruses in our laboratory, but more generally, virus-based nanoparticles, and perhaps nanoparticles more generally, to detect and treat disease. And hopefully we'll get, I know we will get into pediatric-focused diseases, but this subject also extends to adult disease.
Okay, can you tell us what a nanoparticle is for anyone who may not know?
Sure. So a nanoparticle is basically a very small particle, as one would imagine. There are customary size constraints put on the word typically. So a nanoparticle is typically something smaller than 1 micron, 10 to the minus 6 meters, less than the width of the human hair. Commonly, though, we think about nanoparticles as being about 200 nanometers to 10 nanometers in diameter, and that is unimaginably small. I'll tell you that's about 10 to 1,000 times larger than most molecules we think of, and about 1,000 times smaller than a grain of sand. Okay. So there's a range in there that is, as we will discuss, very important in biology, very important in nature, and that's what nanoparticles are.
Okay, and so viruses.
Yeah.
We all know about viruses, right? They make us sick, they give us the cold, they give us the flu. Mm-hmm. But you're talking about using them in a little bit different way today.
Correct.
Yes.
So I want to introduce the audience a little bit to viruses perhaps in a different light. And pardon me for doing this, but I'm going to bring up my computer so I can show you some images. So this is a picture from Wikipedia, which I love, of the influenza virus. And what you're seeing here is those oblong shapes are each a virus particle, a virus nanoparticle. And you won't be able to see it on this image too well, but there are knobs, little protrusions that stick out from the surface, and you can see it a little bit as a halo around each of these viruses. The wavy thing on the bottom right of the image is a cell. And these viruses some of them are interacting with the cells, some of them not. So this is one of the viruses that makes you sick. But let's look at this structure in a little bit different way. This is actually an image from the Centers for Disease Control and Prevention website, the part of their website that deals with the influenza virus. And this is a representation in a cartoon form of the functional parts of a virus. And so the first thing I want you to appreciate, to be honest, is just how pretty they are. Now, in nature, of course, they're not colored red and blue, but they really do have these structural elements, and viruses really are this kind of regular spherical or oblong structure. So in this case, the red and the blue represent different proteins that are on the outside of the virus. Actually, the red ones are the ones against which we raise an immune response to make the seasonal flu vaccine. Okay. So that's the part that will change from year to year. The green stuff on the inside is the DNA, that is the genomic code for the virus. So what a virus is simply a capsule, a bag, if you will, for encapsulating the instructions to replicate the bag. Okay. That's all it is, right? Now, a virus is not a living thing, but is the simplest unit for packaging and replicating a genome that's found in nature. So it turns out viruses are everywhere. They are, in fact, the second largest biomass on the planet.
Wow.
Not us, not mammals, not plants. The first largest biomass, believe it or not, is bacteria, and then viruses. So viruses are everywhere. I want to show you now a family of viruses. So this is just a small snippet of the literally limitless number of viruses that we encounter in our daily lives, certainly in trillions. These are different viruses that have been, the structures of which you are seeing, and they have been determined by various very interesting methods so that we know where all the atoms are on these viruses. So the first thing I want the audience to appreciate is that for a nanoparticle of this size range, and we're in here around 100 nanometers in diameter, viruses are what nature uses to package genes, but from our perspective in the laboratory, they're very useful because we know where all the atoms are. And that means that one of the fundamental connections to make here is the connection of molecular structure, where the atoms are, to the function of the virus or of whatever it is you're dealing with. Since we're in the business of making medicines or making things that can detect disease, anytime we have a platform that we can control the molecular structure of, and if the molecular structure determines the function, we have a really interesting platform on which to engineer different functions to detect or treat disease. The functions of the virus are different than that, but that doesn't mean we can't use them for our own purposes.
Okay.
Right? So here's one more image, and then I'll switch back so we can continue the discussion. This is an early image from our laboratory where we created an artificial, or rather, we artificially elaborated on a known virus to put something on it that didn't exist before and that doesn't exist in nature. So in this image, these little gold spheres you see are in fact small clusters of gold atoms that we arranged very carefully on the virus, and we developed methods to do this as a demonstration that we could take a natural nanoparticle and imbue it with non-natural function.
Okay.
So it's all about function and structure. That's the first lesson. Let me go back now. I'll get out of these slides and return us to the regular image. Whoops. And here we are. Okay.
Okay.
So that's what a virus is.
Okay. And so here, if you're able to control it like you just described, what can you do with it then?
Okay. So the second principle to understand is that in order to identify or treat disease, one has to discriminate between the diseased cell or the thing causing the disease and your cells that are healthy, right? So viruses naturally do this very well, right? Typically, a virus that infects you or infects any mammal doesn't infect every cell in the body. It infects only certain cells for which it carries a key to unlock a mechanism to get into the cell. So viruses are already naturally really good at recognizing one cell and ignoring the billions or trillions of other cells that it may contact in its journey through the world. So that's the first function that we want to learn from. Sometimes we hijack it completely with a virus. Sometimes we learn from the virus how it does this, and we engineer a way to apply that to a different particle. So the first thing we do then with a virus is say, here's a relatively big thing from the point of view of a medicine. It knows how to get into cells, so I'm going to use those principles to recognize and get into the cells I want to get into. That's function number one. The next thing we want to do when we treat disease is to usually or often kill the cell that's making us sick. Sometimes we don't want to kill it, we want to reprogram it or do something else. We can talk about that later. But most of the time, when you take a medicine that is, for example, an anti-cancer agent, you unfortunately have cancer and you want to treat that, or you're trying to get rid of an infection, you want to kill the cells that are either proliferating as cancer cells or making you sick. So a virus is an interesting thing. A nanoparticle in general is an interesting thing because from a chemistry point of view, it's very large. Therefore, there's lots of real estate. There's lots of literally molecular landscape on which I can put different things that have different functions. In the scientific parlance, one way we express this is we call them polyvalent. Valency means ability to grab onto things. Such a large surface with lots of different molecular entities can grab onto cells in ways that are selective for certain cells, but also can grab onto medicines and bring them along for the ride, right? So those are the two essential functions that we learn from when we study viruses in nature, and we engineer when we use viruses or other nanoparticles to create medicines and diagnostic agents.
Okay, and so if you're using a virus to attack something, bring medicine along, for example, are you using the flu virus, or are you using the cold virus, or are you doing something else that maybe doesn't hurt us?
Right, so we do not, and this is true for the field in general, with one exception, which I will talk about, we do not use viruses that propagate in nature, right? So what my laboratory does, and what many laboratories do around the world, is we just take the portion of the virus that is useful to us and leave out other portions of the virus that enable it to propagate and cause disease or just propagate in nature. So in the images I showed you, what those really are just the capsules. None of those images that I showed you have the gene inside that's necessary for the virus to replicate, or some of the other machinery that might be necessary in certain cases. So we call those virus-like particles, but here, or other names, here I'll just call them viruses, and understanding that we use safe versions of them. Now, the exception that I mentioned is sometimes, and this has been an idea that has been in medicine for 100 years, but is really only now coming into full fruition. Sometimes if you know a virus kills something, and if what it kills happens to be what you want to kill to remove a disease, well, then the temptation is to use the virus to kill its natural cell, right? The problem with that, or the warning we always keep in mind, is if we put something into our bodies that can proliferate, maybe something will happen that we don't intend, right? So there are two general areas in which scientists and doctors are pursuing in this general way to attack disease. One is just what I said. If we want to kill a bacterium, there are now many people around the world who are looking at delivering a virus that selectively kills that bacterium and nothing else, and then the virus goes away when the bacterium is dead. That has real high promise. There's some wonderful work going on here at Georgia Tech in that area, not in my laboratory, but it's really quite promising. The other, which is even more promising and much more generally used now, is to use a virus to deliver a gene so that we can reprogram or augment, correct a mistake in the mammalian cell. So now we're using a virus that doesn't get into a foreign cell, doesn't get into something that's making us sick, but it gets into something that can fix the problem. Okay, we'll talk more about that later if you wish. But those are the two ways in which we deliver viruses that have some of the functions of natural viruses. But we really regulate that quite tightly.
Okay. And so how do you make the viruses that you use do what you want them to do as opposed to them doing what they want to do?
Right. So this is an exercise in learning how nature works and applying those lessons iteratively to things we make in the laboratory. So if we choose not to start with a virus that replicates in nature, let's say we start with just this capsule that we're talking about. Now we have to understand how the capsule naturally interacts with cells in our body. That we can study. So that's the first step. Then if it doesn't do what we automatically want it to do, which it usually doesn't, we need to put that function on or into the virus. So that then uses techniques that my laboratory has developed and many others to add to the virus scaffold, if you will, units, new things that will bind to a cell of interest, and separately, or perhaps the same, things that will allow, after binding, the virus to get into that cell of interest. So this requires us to take knowledge from a wide variety of fields in biology to pick the functional units from molecular biology and bring them to our virus particles. So we've developed ways to literally staple